Thanks a lot for the opportunity to speak today. So yesterday, I think we had a chance to do a show of hands for ID clinicians, data scientists, and another group I don't remember. But have we done a show of hands for clinical microbiologists yet? If not, okay, great. So we have some clinical microbiologists here. So I myself am a clinical microbiologist. Maybe I should have titled this Incorporating AI into Clinical Microbiology Practice, because today what I'd like to talk about is some specific technologies that I think are either, or that I know are either in use in clinical microbiology or are very close to becoming in use in clinical micro. Now, I'd like to start just by reminding us about the goals of artificial intelligence in clinical microbiology and grounding us in the fact that we have two sort of constituencies within the clinical microbiology that are invested in the clinical microbiology lab. The first, I think, is the clinician who's looking for actionable data to improve the care of their patients, first and foremost. And to them, this will mean more informative results, ideally more informative results with reduced turnaround times, but also these reports should be easily interpreted, unambiguous, and in a language that they can understand. And of course, the laboratory is interested in all of these things as well, but with the addition of that the laboratory wants their artificial intelligence to lower the cost per test, to reduce the hands-on time of our technologists, and to ideally, if we can, implement these things into existing workflows and make them usable by technologists without extensive additional training. And so then which type of processes are we talking about here? So what type of things are actually going to become in practice in clinical micro in the near future? And I think the first one that I'm going to speak about is something that's already in clinical microbiology practice, and this is antimicrobial susceptibility testing directly from positive blood cultures. I'm then going to talk about gram stain interpretation using artificial intelligence, and then a bit about urine culture interpretation from plates. So this workflow has been explained well by previous speakers, so I'll not dwell on it too much in this case. But suffice to say, the clinical microbiology lab is one of the least automated labs in the hospital right now. And from the time that a clinician selects a specimen from a patient and starts empiric therapy, it takes them at least a couple hours to get any kind of information about the treatment, and that usually comes as the form of a gram stain that allows them to give a best guess about what the appropriate empiric therapy is, but it may not be 100% right. After that, you're waiting another 2 days before you get definitive susceptibility testing results, so you know which antimicrobial will work for the organism that's actually infecting the patient. And in the unfortunate event that you have a highly antimicrobial resistant organism, this turnaround time might be even longer if you have to send to a reference lab for supplemental testing. In our experience, this can take up to a week. And so where I'm going to start in this workflow is sort of right in the middle in the susceptibility testing category, because this is where artificial intelligence, I think, is making a really big impact right now. So just a little bit of background about antimicrobial susceptibility testing. And typically in many labs, this is performed by automated systems, which, as a bit of an aside, if you take the definition of artificial intelligence in sort of the most simplistic terms of a computer making decisions, these systems already have artificial intelligence built into them because they already know which antimicrobials go with which organisms, and that you shouldn't report, say, vancomycin for an E. coli. The images here are just some of the pictures of the automated susceptibility testing systems in common clinical use. So one of the limitations of our current susceptibility testing methods is that they require isolated colonies that are growing on agar. So that means if you have a blood culture, you have to wait for that blood culture to turn positive. You have to grow that up on a solid medium. And then you can perform antimicrobial susceptibility testing on the colonies that you get. And the total turnaround time for this is 36 to 48 hours if everything goes perfectly, which of course in clinical practice, everything often does not go perfectly. So that means that our current antimicrobial susceptibility testing method, although it works well for many specimen types, is simply too slow for supporting treatment of rapidly progressive infections such as sepsis. And why is this? Is basically because this requires organism growth for readout, and just that growth itself takes 12 to 24 hours of incubation. And these susceptibility testing methods have a limited detection, right? So you inoculate a certain amount of organisms, and you don't really start detecting growth until somewhere in the ballpark of maybe 1 times 10 to the 7, so 10 million organisms per mil. So how can we make this better? We can't make organisms grow faster. We can't, alter the biology of these organisms, but we can develop better detection methodologies. And conceptually, the fastest way to detect organism growth is simply to look at the organisms on a single cell or several cell basis using microscopy to monitor their behavior in the presence of antibiotics. Now, that sounds great, and it seems like, oh, sure, we should have been doing this for a long time. But in reality, you can't simply set up a microscope and ask a technologist to watch a bacterium grow and ask you if it's susceptible or resistant. And this is where artificial intelligence really comes in, because until we had artificial intelligence image interpretation, we couldn't perform these types of tests. So this is a currently existing system that's cleared in the United States. This is the Accelerate system. That performs microscopy-based susceptibility testing directly from a positive blood culture. So it works by having a technologist load the organisms directly into a cartridge using a regular pipette, and this is directly from a blood culture. And the organisms are then incubated in the presence of an antimicrobial and monitored by automated microscopy. And you can see in these images, the top image is an E. coli strain that's growing in the presence of an antibiotic and that it's susceptible to. So you can see some really classic morphologies here. Is the E. coli cells are filamenting. You see a central bulge. That's classic for inhibition by a cephalosporin or a beta-lactam antimicrobial. Now, in the bottom image, you see something that's qualitatively very different. So this is not single cells you're looking at. Now you're seeing microcolonies of cells. They're increasing in biomass, and this is another E. coli strain growing in a antibiotic that it's resistant to. And so within this system, there's an AI model that is essentially locked. It's basically a black box that predicts susceptibility based on correlating these image data with known libraries of susceptible or resistant bacteria, and provides a antimicrobial susceptibility result within 8 hours. Now, this sounds really great in practice, but I'd like to also mention a couple of limitations of these types of systems. And I think the biggest limitation, and this is something that I think was brought up in a question yesterday, is that in some cases, molecular methods are actually better at certain applications. So I think a great example is that most resistance in gram-positive organisms can be predicted by only a few genes. So for example, if you have a Staph aureus and you use molecular detection and you find the MEK-A gene, then now you know you have an MRSA, and that actually tells quite a lot of the story that you need to know. So in a lot of cases, these artificial intelligence-based systems are best applied to gram-negative bacteria, where genotypic prediction is not quite as straightforward as in gram-positives just yet. And another one of the limitations is that they're simply an order of magnitude more expensive than standard methods. So they're currently limited to large, well-funded academic centers, typically. So that leads us to actually a question that our lab was addressing is, so how can we take sort of this underlying technology, this artificial intelligence based susceptibility testing, and apply it to smaller centers with fewer resources, and how can we take this and make it a little bit sort of conceptually similar? So our lab actually worked on developing some off-the-shelf rapid diagnostics by combining inkjet printing technology with automated microscopy and deep machine learning. So we start by using this commercially available printer that's produced by HP that's basically designed to accept whatever solution you want and print it out into the wells of a microtiter plate. So in our case, we load these little cartridges with antimicrobials and prepare dilution series, just like a standard broth microdilution panel, within the wells of a 384 well plate that are actually coated by a microscopically compatible solid surface so that the cells are sitting in a nice layer that we can focus on. So after we sort of dispense these droplets of antimicrobials, we develop the concentration gradient, which is then overlaid by bacteria, which are also dispensed using the inkjet technology here. So really what we've done is we've developed not only a microdilution series, but also we've inoculated that simply by doing 2 pipetting steps, which this process takes many, many steps to do manually and is well outside the technical capacity of most clinical laboratories. So after we inoculate the cells, we can then incubate them, and in this case we incubate them for 2 hours, and monitor by automated microscopy. And that's what this image on the right shows. So in panel A, this is what an E. coli strain looks like after it's been treated with ciprofloxacin. So it fails to divide, and it just elongates and gets longer and longer, and eventually will die. Panel B is a cell that's filamenting and has a central bulge, and that's being inhibited by a cephalosporin. Panel C is kind of boring because it's just sitting there doing nothing, and because it's being inhibited by gentamicin, which is a protein synthesis inhibitor, doesn't change the cell morphology, just kind of stops it dead in its tracks. Finally, panel D is meropenem. That's the classic morphology of a cell inhibited by a carbapenem, is one that just balloons up to many times its normal size. So what we did was we took these images and took the knowledge that organisms change their morphology under different antimicrobial pressure, and then trained a deep convolutional neural network to recognize these inhibitions and correlate the images to a degree of inhibition, and that's what's shown in this panel here. So the red is showing inhibited regions of the image, and the gray is showing areas that are growing. And we can then graph this, and we can have on the x-axis concentration of antimicrobials, and on the y-axis degree of inhibition, and we can then call minimal inhibitory concentrations based on those data. So we compared this to the reference standard, which is broth microdilution, and we found greater than 95% concordance with this standard for a subset of organisms, which is considered acceptable for various regulatory agencies. Now, what we're doing in the lab currently is we're trying to work to make this convolutional neural network a little bit simpler. We're trying to improve our algorithms for detecting the bacteria and making this whole process a little bit simpler and potentially more implementable in a clinical laboratory. And so what I wanted to talk to you about next was a different step in the diagnostic process that we work on. So we're taking a little bit of a step backwards now, and now we're going to move on to staining and microscopic analysis, which is really important for guiding the initial therapeutic decision for treatment of an infection. And just a bit of background on that, and the gram stain is pretty important in that it provides a general sense of what type of organism is present. So you can get a result like, gram-negative rods, gram-positive cocci in chains. So gram-negative rods are pink, gram-positive cocci are purple, or gram-positive cocci in clusters. And each of those imply a very different type of treatment. And what's, I think, important for artificial intelligence is a limited number of bacterial morphologies constitute the overwhelming majority of pathogens that are of interest. But one of the limitations of applying artificial intelligence to a gram stain is that gram stains are inherently very noisy. And this is an image that we took of a gram stain that was prepared during the normal course of clinical practice in our laboratory. So for all the materials with our gram stain project, we simply used things that our technologists prepared, so we wouldn't be biased towards, perfect gram stains. These are things that are actually being used in clinical practice. So this is a very noisy image, and you can see that there's stain crystallization. There's a lot of background artifact that's likely resulting from the material in the blood culture bottle. And if you can appreciate it, you can start to see sneaking around in here little gram-negative rods. So that makes interpretation a bit difficult. And I would suggest that, like, I would love to just implement a simple thresholding method and say pick out everything that's the appropriate shade of pink or red, but that becomes very difficult with all this background in the images. So we trained a convolutional neural network using 100,000 images that represented 180 slides to recognize the different bacterial morphologies that I talked about previously: gram-negative rods, gram-positive cocci in chains, and in clusters. And when we look at the data for our classification, we can have a excident area under the rock curve here, and we have good sensitivity and specificity for gram-negatives and gram-positives in clusters on a whole slide basis. So if we look at with many images and then decide what the predominant organism is, we do a great job, I think, for these organisms. So we have a greater than 95% sensitivity and specificity for gram-negatives and greater than 93% for gram-positives. The algorithm still struggled a bit with gram-positive cocci in chains, and I think that's likely due to, partially at least, because of the fact that gram-positive cocci in chains, when you prepare a slide in clinical practice, often will clump together and sort of look a little bit like clusters. So what we did was we performed some data augmentation on our images, and this is something we did after the publication, and we were able to rotate the images, change, flip the images, greatly increasing our data set size, and we were able to achieve a greater than 99% classification accuracy on a per image basis. So we were able to classify these simple morphologies of gram stains relatively accurately, but that's really not the whole story, because interpretations of gram stains are not really always that straightforward. So this organism here, it looks like it may be sort of living somewhere between a gram-positive and a gram-negative. This is actually a gram-positive lactobacillus. To sort of the untrained eye, you might think this is gram-positive cocci in clusters, but it's really a mycobacterium that's staining poorly, which, if this was misinterpreted as gram-positive cocci in clusters, would have significant clinical impact. This thing that kind of looks to me like maybe a robust gram-negative rod is actually gram-negative diplococci. So reports in clinical micro can vary pretty significantly based on technologist experience, time spent per specimen. Now, I remember someone said that previously that AI is not as good as pathologists who have an unlimited time, and our technologists definitely do not fall under the unlimited time category, because they can only spend a very short amount of time per specimen because of our volume. Stain quality and specimen type can also impact this, and I think I would just like to sort of illustrate this with an example here. This is a gram stain from an aerobic blood culture bottle, and these are large organisms, gram-positive staining. They have buds, so that's a yeast, right? So a inexperienced technologist may simply call this a yeast, and the clinician may interpret that as a candida. I think that's not unreasonable to think that sort of any clinician that you, like a random clinician in the hospital, may think of yeast, think of candida. A more experienced technologist may call this budding yeast, probably still going to be thought of as candida. A experienced technologist may recognize that there's a little bit of clearance around this organism that is actually representative of polysaccharide capsule, and they may call this encapsulated yeast, which probably gets a little bit closer and starts adding Cryptococcus to the differential. But the most experienced technologist would be able to call this encapsulated yeast suggestive of Cryptococcus, which definitely forces the issue and has the clinician consider Cryptococcus in this case. And indeed, this is a Cryptococcus case. So wouldn't it be great if we could have all of our most experienced technologists all of our technologists be able to call this organism as a Cryptococcus, where most of our technologists may not be as comfortable doing that. And that's, I think, where AI will be going next in this space, that we could have basically an unbiased interpretation of gram stain, maybe with a confidence associated with that. So we have 92% confidence. Crypto, it's probably not candida. If you're thinking about it, this may be hidden from the clinician because that may be just too much information. So I think this is an interesting next step. And to that end, we actually sort of try to pilot that on our data set by creating a t-SNE plot of our gram stain data, actually. And we could see that if you sort of zoom in on the images that are represented in this little cloud here, we can see some shorter gram-negative rods, maybe with a little bit of background noise. But if you look in different parts of the plot, you can see qualitatively different gram-negative rods with qualitatively different backgrounds, which suggests that our AI may be able to provide more nuanced information in the future about gram stains, and we're looking forward to exploring that a little bit further. So finally, I'd like to talk about a little bit of limitations of this, because actually, ironically, one of the limitations of an AI-based gram stain is that technologists are really good at this. Technologists have greater than 99% concordance with culture results on the gram stain at this point, and our AI is in concordance with trained personnel about 92% of the time. So in addition, computers probably make different mistakes than a human, and this is kind of I think this is a neat little image that I pulled out of a Wired article that basically fed a neural network some unusual looking images and saw what it was thinking. So for example, this in the top left corner is an image that to us looks sort of like nothing, but the algorithm is calling it a king penguin. So I will not call myself a zoologist, and I think I'm going to the zoo tomorrow. I may learn more about this, but I think this is not a king penguin. This is not a starfish, right? So our humans would not make these kind of mistakes. So humans will never mistake this, but an AI might. And fine, it doesn't really matter if this is called a king penguin or what, but in gram staining, this error could be life-threatening. So misclassification of a gram negative for a gram positive could result in, say, inappropriate administration of vancomycin and adverse patient outcome. So it's very important that we may consider the limitations of our algorithms and potentially get technologists involved as well, and try to combine the power of our technologists with the power of our artificial intelligence to speed up their classifications of gram stains rather than completely supplant it. Okay, so last part about AI algorithms here, I'd like to go to a little bit about culture, and I think this has been reviewed by a couple of other speakers, and I think it has been because I think this is a really important future application of AI, which is reading culture plates. So a lot of clinical micro is still based on interpretation of colonial growth on plates, which tells you the relative amount of viable organisms and allows for our identification and susceptibility testing. And these are just sort of representative plates of some E. coli's and some Staph aureus that we could see in the clinical lab. So plate interpretation can be a bit subjective, and colony morphology is often used as a initial screen. And this is an image from a paper here that basically shows a mixed culture of a variety of different types of organisms. And I think an illustrative question here is, how many microorganisms are represented by the plate to the right? And the question is, or the answer is, it would take me some time to figure that out. And I think if we sort of got together and discussed it a little bit, we could probably figure how many are there. But if you asked 4 or 5 people their opinion, you may get different answers. So AI might help us allow us some unbiased interpretation of these colony morphologies in a quicker way than we normally might. So what I think is also important is that many or even most plates in clinical practice have no growth, which is a great opportunity for AI triage, which saves on our technologists time and prevents and allows them to focus on working up some more impactful cultures. And I think this is particularly important in urine cultures because there's very high volume. Interpretation really is a burden on technologists. We implemented a reflex urine culture where we only reflex urines that have positive urinalyses, and we still have a lot of negative cultures. And the technologists, they didn't sign up to just triage negative cultures. They want to be impacting patients by working up organisms, really. And this really sort of contributes not only to excess cost, but also some fatigue and dissatisfaction amongst their technologists. And I think they're particularly amenable to AI too, is because not only are they often negative, but there's only a few pathogens represent most of our infections. At least in our practice setting, we can probably list off the majority of the pathogens. And interpretation errors, unlike sepsis, they're usually not life-threatening in this case. So that sort of leads me to the question: is if we have all this opportunities to perform AI, and why has AI not yet sort of caught on in smaller, less resource settings? Now, I think here we have a bit of a bias towards having AI in our labs because we're larger academic settings. But if you go to sort of random hospital in the United States, you're less likely to find artificial intelligence. And I just wanted to quickly go through the some of the reasons why I think that's the case. And one of the biggest ones is literally space. So in order to digitize information, you often need automation, and microlabs simply have not been designed with that in mind. Also costs a lot of money, and a model that we often use in the microlab, this reagent rental model, where we purchase a certain amount of reagents and then lease the instrument from the company, may or may not work here if there are no reagents. Technologists may not be trained yet to include AI in clinical practice, and the question of whether we need informatics support, I think, is probably obviously yes, that may or may not be present in the hospital. There are some administrative issues, such as LIS systems not being able to report complex data, such as images. Now, of course, we have EMRs that report images, but does our LIS maybe or maybe not? And then how does one verify the performance of these AI methods? And I just quickly like to go through a couple of these. The first is space, and we need automation to digitize data, but we haven't quite got all of the automation down yet, and mostly because we get a huge diversity of specimen types in micro. So any given day, our lab could receive any or all of these specimens, anything from an explanted knee to a bioprosthetic heart valve to something much more easy to deal with, like maybe a swab. So standardization of specimen collection and implementation of automated processing systems would be really important in the future for digitization of AI. So luckily, there are some systems that exist, and we're coming hopefully ever closer to fully automated microbiology labs, especially as labs get upgraded and start thinking about these things. Now finally, how does one perform verification? For AST systems, it's really easy. Performance metrics are already established. You can compare them to manual methods and run QC just like everything else. So these slotted into the clinical microbiology laboratory very nicely, and I think these are relatively straightforward. But for plate or slide reading, we have to start comparing the error rate to a technologist, think about what are our routine quality control and quality assurance requirements, and when do we have technologist review of positive and negative results? My sort of worst case scenario is what if the instrument starts, especially with adaptive algorithms, starts misclassifying plates and we are not reviewing them, and all of a sudden things start getting released inappropriately. So this requires a lot of resources to implement. So to conclude, I just want to sum this all up in sort of a simple way, I think, that says AI can make the most impact in clinical micro when it's used to augment or replace, in some cases, commonly performed manually and experientially demanding processes, such as gram staining or triage of cultures. It can also, of course, add additional utility, such as looking at positive blood culture susceptibility testing. I think what's really important too is the complexity of an algorithm is not necessarily related to clinical utility, because I think the most impactful and widespread AI is the expert systems on susceptibility testing methods that tell us what to report for which organisms. And these things have made huge impact by simply just being a list of rules that the machine follows. And of course, that any novel test methods really should be designed to support clinical decision making, and to do that, we need to definitely have collaboration between not only clinicians but computer scientists. So just to conclude, I'd just like to thank people in our lab and then some institutions and companies that provided financial support or material support for our work, and I'll take any questions.